So we've explored the properties of enzyme active sites, how they provide a precisely shaped environment for the substrate, position substrates in the right orientation, and can help stabilize transition states. So now we're going to look at how enzymes actually use these features to make reactions happen faster. We're going to talk about the active site as a dynamic environment where the side chains of the amino acids in the active site are going to play a role in activating or lowering the activation energy and driving a reaction forward. So there's three main classes of strategies that are used. Acid-base catalysis, where amino acid side chains act as proton donors or acceptors. There's covalent catalysis, where the enzyme temporarily forms a covalent bond with the substrate. And the key word there is temporarily, because a key feature of catalysts, and enzymes are catalysts, is that they can be reused. So if you form a covalent bond with the substrate, you're going to have to be able to cleave that covalent bond to recycle and regenerate the active site. And then metal ion catalysis is where a bound metal ion cofactor helps stabilize charges. So if we have a positive metal ion, it's going to help stabilize a negative charge somewhere in the course of the reaction. Or these can participate directly in an electron transfer. So remember that we talked about metals as being, redox helpers. They participate in redox reactions. Okay, so again, remember that these are catalytic strategies. We would call them enzyme mechanisms, but there's a difference between an enzyme's mechanism, which is a catalytic strategy, and a reaction mechanism that's a class of organic reactions. So first, we'll talk about general acid-base catalysis. So general acid-base catalysis, again, is going to involve a molecule besides water. So that's where the general comes in. Okay, so if water acts as the proton donor or acceptor, we would call it specific acid-base catalysis. So when water is the proton donor or acceptor, it is specific acid-base catalysis. But in this case, we're talking about amino acid side chains in the active site, and so we're considering general acid-base catalysis. So this is going to be limited to our ionizable amino acid side chains. So remember, you have to be in the acid form or the protonated form. So let's label these more specifically. Your general acid form is when your molecule is protonated, and so it can give off a proton. And the deprotonated state would be the general base form. So this is the two forms you can have and the roles they can play in a reaction. Okay, so if you need a proton donated or one removed, these would be your players. You have histidine, aspartic acid, and glutamic acid, serine and threonine, tyrosine, cysteine, lysine, and arginine. Now, some of these are more likely than others, right? So, and that's based off of their pKa value, right? So the ones that, are really very common is histidine, and serine often will play a role, and aspartic acid and glutamic acid. So those are the ones that kind of are most frequently are acids, and then lysine and arginine tend to be the bases that are more frequently used, okay, because of their higher pKa's. But remember, the pKa can change in the active site. All right, so let's look at an example of acid-base catalysis. and see how we can recognize it and how it actually works. So, again, it's going to involve either water, which would be specific acid-base catalysis, or a functional group, which would be general acid-base catalysis. So we're going to look at the example of pancreatic ribonuclease. So in this case, okay, pancreatic ribonuclease serves the role of cleaving. It breaks the bonds in an RNA molecule. Okay, that's hence the ribonuclease part. It's going to use two histidine residues that function in a general acid-base mechanism. So we're going to zoom into this, and it requires the addition of water as well. All right, so here we go. So we have our two histidine residues that act in this mechanism. So we've got one here, okay, and then we've got our second histidine here. Okay, so those are our histidine residues. And let's look at what histidine 12 does first. So histidine 12, notice that we're using our curly arrows like we do in organic chemistry to show where electrons are going. So how are we going to recognize that histidine 12 is acting as a general base? First, we're going to recognize that it starts out in the deprotonated state. Okay, so this starts deprotonated. And then we're going to focus on the arrow, right? So we've got our lone pair there. It's going to form a bond with the proton. And what we do is we're going to look at the next state that we find our histidine in, and now it's protonated. So if my molecule goes from deprotonated to protonated, it has acted as a general base. Okay. Now, if we look at our His 119, we'll notice that it starts out protonated. And then when we look at it over here, okay, it has been deprotonated. That means it has given up a proton. So His 12 is going to act as a general base to take a proton from the RNA molecule. Okay, so that is on the hydroxyl group on position 2. So remember how we said that RNA molecules were liable to have their backbones hydrolyzed in basic solutions, and that was due to this hydroxyl group here that does not exist in DNA. So here we are, the base in this case is coming from the histidine residue. It's taking this proton away, okay, and then that causes the oxyanion to attack the phosphodiester bond here, right? And the histidine 119 is going to act as an acid. It's going to donate a proton to the RNA molecule, okay, to help it wind up leaving the active site. So histidine 12, it's going to abstract that proton. Then that's going to wind up causing the cleavage of that phosphodiester bond, okay? And now what happens is the histidine 12 is now going to act as an acid, okay? And it's going to give up a proton. So if you'll notice at the end of the cycle, okay, it ends deprotonated. So it ends in the same state that it started in, okay? And a similar thing is going to happen to our His 119. So a water molecule is going to enter the active site, and the His 119 is going to be a general base. It's going to take a proton from the water molecule, and that's going to leave this as a hydroxyl. That hydroxyl is going to attack the phosphoryl group of the remaining RNA, and that's going to allow for product release, and the histidine residues are going to wind up being in their initial state. So it's going to end protonated. Okay, so that's how acid-base catalysis would work. So now we're going to look at covalent catalysis. So in covalent catalysis, okay, you're going to have a bond forming, as we mentioned before, between the enzyme and the substrate. That's going to involve a nucleophile on the enzyme, which is going to attack an electrophile on the substrate. And that's going to form a covalent bond between the enzyme and the substrate. And eventually, in the cycle of the reaction, okay, so by the end of the reaction cycle, this bond will be broken to regenerate the enzyme itself in its initial catalytically active state. So we're going to look at the example of glyceraldehyde 3-phosphate dehydrogenase. This is one that uses a coenzyme NAD+, and the sulfhydryl on a cysteine, which forms a covalent bond with the enzyme to form what we call a thiohemiacetal intermediate. Okay, so here we go. We're going to zoom in again on our reaction, and we're going to start by noticing the substrate comes into the active site, and the fact that the substrate binds in the active site, okay, winds up changing the conformation such that it's going to begin the formation, okay, of our thiohemiacetal intermediate. So here is, if we look at it, here is our thiohemiacetal intermediate. Okay, so we started with this substrate molecule here. It is now bound as, it's now bound as a thiohemiacetal in the active site, and our covalent bond is this one right here. All right, so now in the next step of the reaction, the NAD+ is going to oxidize the thiohemiacetal back to an acyl, what we call an acyl thioester intermediate. So this here is our acyl thioester intermediate. Okay, so notice that in this part of the reaction cycle, the NADH has been reduced. So the reduced cofactor no longer has as high affinity for the active site, so it's going to leave, but NAD+ is going to come back in. That NAD+, okay, is going to stabilize the negative charge on the carbonyl that is going to develop during the final step of the reaction. So the final step of the reaction is going to involve inorganic phosphate attacking. So the inorganic phosphate, which we always abbreviate P sub I, this is going to attack, the enzyme-bound intermediate, and that's going to cause cleavage of the bond between the enzyme and the substrate. Let me do a highlighter so that appears better. So this is the bond that's going to be cleaved. Here's our product, and our enzyme has been regenerated in the initial state. Okay. And so things to notice that in addition, we've got this histidine in the active site that gets protonated throughout the course of the reaction, right? It grabs the hydroxyl proton over here, and then eventually it donates it back to help cleave that bond and release the product and regenerate the sulfhydryl that was on the cysteine in the active site that was involved in the acyl enzyme intermediate. All right, so that's covalent catalysis and how we can, again, get those common organic reactions that happen and form a covalent bond between the enzyme and the substrate. Okay, so our third catalytic strategy, okay, is metal ion catalysis. So let's look at a case where metals are going to promote proper orientation of our bound substrates, and they can also aid in redox reactions. Okay, so our example of this is going to be carbonic anhydrase, and it's going to use a zinc ion to support a nucleophilic attack on carbon dioxide. Okay, so this is carbonic anhydrase. You've heard of this enzyme before, right? So it takes CO2 and it produces the bicarbonate ion, right? And so this is going to be part of that hemoglobin regulation process where we wind up taking CO2, which is less soluble, and making a more soluble product, but it winds up reducing the pH of the solution in as well, and that promotes unloading of oxygen from hemoglobin, right? All right, so let's look at this cycle, okay? So here we have, in the beginning of the active site, we have a zinc, and remember that what's good at binding zinc? Histidine and cysteine side chains. So the way the zinc is held in the active site is by coordinating to histidines. Okay, now, we also have this zinc binding to a water molecule, and that is going to lower the pKa of the water molecule. Okay, so pKa of water is lowered, right? Okay, so the next thing that's going to happen is because the pKa of this is lowered, it's going to be easier for it to be deprotonated. And so, of course, it gets deprotonated, leaving us with a hydroxide anion. So there's the hydroxide, it's got a negative charge. Okay, so now this is bound in the active site, and it's the water is held in the active site by the zinc. Okay, so let's take note of that. So the deprotonated water, otherwise known as a hydroxide ion, is held in the active site by the zinc. Okay, and so now what happens is this hydroxide can do the nucleophilic attack, right? So here's the carbon dioxide, and over again in this course, I am going to, and I'm going to make this a little smaller so it's easier. I'm going to remind you, right, that our carbon and our oxygen bond is polar, whether it's single bond, double bond, whatever, because oxygen is more electronegative, okay, than carbon is. Okay, so that is going to make this carbon dioxide, the carbon in the carbon dioxide, an even better electrophile or a site for a nucleophilic attack. Okay, so now we get the nucleophilic attack on the carbon, right? And so that leaves us with this bicarbonate product, but it's still coordinated to the zinc. Okay, so it's, but if you'll notice, okay, This now has positive charge, just like the zinc ion has positive charge. And so that buildup, okay, is going to put strain on this bond. And when another water molecule comes in, it's going to be more favorable for that water molecule to displace the bicarbonate. All right, so the zinc, okay, so what's going to happen is a second water molecule is going to come into the site. It's going to pop the product off, and it's going to have another water molecule come in, right? So here's our product. And if we zoom out and we look at what our enzyme looked like when it began, right? Where we ended with a water bound to the zinc is where we began, with a water coordinated to the zinc. So that's how this uses the zinc, okay, to make this a better nucleophile, allows this reaction to happen more easily, and then another water comes in, knocks off the product, and gets in place for the next cycle of reaction. Okay, so those are the strategies, okay, that we can use for catalysis. And so now we're going to talk just a little bit about the types of reactions that we can get. So we can have coenzyme-dependent redox reactions, metabolic transformations, or reversible covalent modifications, right? So you've heard about these reversible covalent modifications before, right? So I will often call them post-translational modifications, right? And we use these to regulate the activity of proteins. Modifications. And the one that we've mentioned most frequently is phosphorylation. Phosphorylation. All right, so let's look at a few of these. All right, so coenzyme-dependent redox reactions. So again, redox often requires coenzymes because, again, they can carry electrons. With our NAD example to NADH, NAD plus to NADH gives us, carries, remember it carries 2 electrons, right? And then FAD is the oxidized form of what is a 4-electron carrier, right? Because we go from FAD to FADH2. So that's a 4-electron carrier. All right. So again, NAD, NADP, FAD, and FMN. So FAD is flavin adenine dinucleotide. FMN is flavin mononucleotide. NAD is nicotinamide adenine dinucleotide. And NADP is the phosphorylated form of that. So here are some examples. Again, dehydrogenases. That word is an indicator to you that redox is going to be happening, okay? And redox using NAD, okay? All right, so here we have an example of taking pyruvate, and the pyruvate gets reduced to lactate, right? So we're reducing here we're adding hydrogens. And the thing that gets oxidized, the source of our hydrogens and associated electrons, is NADH. Okay. All right, and in this case, where we're using FAD, okay, we are taking succinate and we're going to oxidize it. So we're going to be making a double bond between those carbons and removing those protons or the hydrogens and their electrons. And so we're going to wind up in the end with fumarate as the product, and we've passed electrons on to FAD to make FADH2. Okay, so I think I made an error here. Boop. So in this case, Nope. Never mind. All right. So then the next class of reaction we're going to talk about is metabolic transformation reactions. Okay. So these are essential components of anabolic and catabolic pathways. So remember, catabolic is breaking down of molecules, and anabolic is building molecules. So these metabolic transformation reactions are going to be things like isomerizing things, so changing the connections or stereochemistry. We're going to be having condensation, so joining molecules to build bigger molecules, and things like dehydration or hydrolysis reactions, so either removing or adding water to break things apart, put them together. So a lot of times in biochemistry, we often combine substrates with the same molecular formula, so we're making polymers. So I'm just gonna, that's what we're referring to here. Okay, so all right, so these are things like triose phosphate isomerase, which is an enzyme that takes dihydroxyacetone phosphate, all right, and it rearranges it to form glyceraldehyde 3-phosphate, right? And then when we're doing things like building lipid molecules, right? We have things called something called prenyltransferase that's going to help add these alkyl groups onto our substrate. All right, and then the last class of reactions, types of reactions that we're going to talk about are metabolic transformation reactions. Okay, so these again are our post-translational modifications that happen to regulate activity. Okay, so this is the key word here is it's used for regulation. Okay. And it's going to be turning on or off signaling or gene expression. So it's going to be turning things on or off. So kinases are things that add a phosphate, and phosphatases are things that remove phosphates. The site that they use, the sites that they add phosphates to will, again, generally have hydroxyl groups on them. So if we look at this example that's on the screen, we've got phosphatidylinositol 4,5-bisphosphate, and where are we adding the phosphate group? We're adding it onto that hydroxyl group there. Okay, so in this case, insulin, okay, when insulin is present, okay, it leads to the stimulation of a phosphatidyl or phosphoinositide 3 kinase activity. Okay, so insulin turns this enzyme on. Okay, and that enzyme then phosphorylates, okay, this substrate right here, all right, and it generates this product here. Okay, then what happens is when in the presence of phosphatase and something called phosphatase and tensin homologs. So this is P10. All right, that can turn the system off by removing the phosphate. Okay, so kinases, use ATP as a phosphoryl group source, and so you'll notice that it's here as one of the factors that helps promote this reaction. Okay, so that gives you an overview of kind of reaction mechanisms. We'll do practice questions in class. And in class, we're going to go through some detailed enzyme reaction mechanisms. And we're going to use chymotrypsin, enolase, HMG-CoA reductase, and fumarase as our examples. So I wanted to set the stage with this, and then we'll talk about things in detail in class.